All right, hi science students. This will be your SOL review lesson number 7. It's about DNA, mitosis, and meiosis. So the key vocabulary you need to know starts with replication. Replication is to make a copy. Mitosis is the process of making copies of cells. So replication is to copy DNA. Mitosis is to go from one cell to two cells. Sexual reproduction is when organisms reproduce using sex cells, like sperm and egg. A chromosome is a bundle of DNA. The DNA is condensed into this shape called a chromosome. Match means to go together, like the base pairs, the matching nitrogenous bases inside DNA. Adenine matches thymine, and cytosine matches guanine. A to T, C to G. The nucleus is the organelle that protects the DNA in eukaryotes. You carry a nucleus in all of your cells to protect your DNA. Cytoplasm is the gel that fills the inside of a cell. It's mostly water. Ribosomes are the organelles that build proteins. They're very small organelles that build proteins. A protein is a large molecule with a specific job inside the body, like an enzyme or to build cellular structures. A sex cell is a cell used for cellular reproduction, like sperm or egg. A gamete is the same thing. It means the same thing as sex cell. Meiosis is the process of making a sex cell, like a sperm or an egg. A diploid cell is a cell with a full set of chromosomes. That's 46 chromosomes in humans. Haploid is a cell with half of a set of chromosomes. That's 23 in humans. So in haploid, you only have half as much. Diploid, you have 2 copies of every chromosome. They're in pairs. A fingerprint is the pattern on one's fingertips that are unique to them. Like I have a different fingerprint than you, or my brother, or my mom. Related means you're connected to one another. So I am closely related to my brother. DNA is the molecule that all living things use for directions to build their proteins. The inside of DNA is made of 4 nitrogenous bases that store all of its information. Adenine matches to thymine, guanine matches to cytosine. You can use a special process called gel electrophoresis to separate pieces of DNA and make a DNA fingerprint. This is the complex picture. This is the simple picture. I'll go through this. So gel electrophoresis happens in this machine. You take your DNA sample and then you cut it into little pieces with special enzymes called restriction enzymes. And you put all those little pieces of DNA in one end of the gel. It's like a Jell-O mold. Then the DNA segments are loaded into these wells in the gel. And then we're at step 3. An electric current is passed through the chamber, and DNA fragments move towards the positively charged end. So you turn on the electricity, and the DNA, like, how can I show this? The little pieces of DNA go, and some will go very far if they're small pieces, and some will take longer because they're too big. So they're like, but the small pieces are like whoop to the other side. So anyway, everyone's DNA will make a different pattern. So here is like one person, and you can see they must be closely related to suspect 2. So if this was the DNA from the crime scene, there was a crime. Who did it? Looks like suspect number 2 did it. 1 and 3 are different. but number 2 matches. So they are the criminal. You can use a DNA fingerprint to determine how closely related different organisms are. You can see that only kitten 4 is related to the cat. So here's a cat's DNA fingerprint. And then there are 4 kittens. And which one is related? Well, 4 is the most similar. You can see this bar is different. That bar must come from the father's DNA. Okay. When a cell makes new proteins, it copies the directions in DNA into mRNA in the nucleus. That's right here. So this is called transcribed. This DNA gene is transcribed in mRNA here. Okay. The mRNA leaves the nucleus and goes to the ribosome in the cytoplasm. That's what I see right here. The mRNA is very small, so it can leave the nucleus and go to the cytoplasm to find a ribosome. Here's the ribosome. The ribosome reads 3 bases at a time to build a protein out of amino acids. See, it's reading 1, 2, 3, 1, 2, 3, 1, 2, 3. Those are called codons, if you care. And then it brings these amino acids, and then a whole chain of amino acids will form a protein. Now, sexual reproduction requires sex cells like sperm and eggs. Gamete is another word for sex cell. Human men make sperm in their testes. Human women make eggs in their ovaries. So these are the ovaries, and these are the testes. They make sperm, ovaries make eggs. Meiosis makes sex cells. Meiosis begins with 1 diploid cell, 46 chromosomes, and ends with 4 haploid cells, the 23 chromosomes. Cells with a full set of chromosomes, like this, are called diploid. Cells with a half set of chromosomes, like these, are called haploid. Those would have 23 chromosomes in humans, 46 in humans. DNA can make a copy of itself by doing DNA replication. DNA replication only happens before a cell needs to divide to make new body cells during mitosis. So in one cell, you have 1 copy of all of your chromosomes. So you have 46 chromosomes. Well, but I want 2 cells. I want to grow up. I need more cells. So now my DNA will replicate in prophase. It will make another set of DNA. So you actually have 92 chromosomes. That's too many. So then you go through metaphase, anaphase, and then telophase, and now you have 2 cells after cytokinesis with 46 chromosomes, 46 chromosomes. Mitosis has 4 phases. In prophase, right here, DNA replicates. In metaphase, the chromosomes line up in the middle of the cell. In anaphase, the chromosomes are pulled apart. And in telophase, the cell divides in half to make 2 new body cells. They're diploid. Mitosis makes body cells. Mitosis begins with 1 diploid cell with 46 chromosomes and ends with 2 diploid cells with 46 chromosomes. Example question. Let's fix that. The process of DNA replication is necessary before a cell A makes a protein, B codes for RNA molecules, C divides into 2 cells, or D modifies lysosome enzymes. Well, to rephrase, why is DNA replication necessary? To make a protein? No, we can just transcribe the DNA into mRNA. We do not need to replicate DNA. Codes for RNA molecules? No, that's just genes. Divides into 2 cells? Well, yes, if I want 2 cells, I need 2 sets of DNA for each of the cells. Modifies lysosome enzymes? No, the Golgi apparatus can do that. The Golgi body is for that. So the correct answer is C. Now I'm going to predict that this question must be about DNA fingerprints and which organisms are most closely related. The picture shows a segment of DNA from a cat. Which of these is most likely the kitten of this cat? To rephrase, which kitten is the cat's child? Well, we talked about this earlier, so what's the correct answer? It was 4, right? That is the most similar. Yeah, it's 4. I'm looking at it again. It's 4. Okay. The parts of DNA that provide the code for proteins are the sugars, phosphates, hydrogen bonds, or nitrogenous bases. Well, to rephrase the question, what parts of DNA have the directions for proteins? Well, I kind of want to say genes, but that's not one of the answers. So what is DNA made of? Well, it has all of these things in DNA, but what part has really the information? That's going to be the nitrogenous bases. That's your A, C, G, and T. Adenine, thymine, guanine, cytosine. That's the special part with information in the base pairs. So the correct answer is D, nitrogenous bases. The main reason that eating salty foods makes a person thirsty, well, I'm just confused. This slide belongs in a different PowerPoint, but whatever, I'll read it again. The main reason that eating salty foods makes a person thirsty is that additional fluid is needed to C, maintain the fluid balance in the cells. You'll see that in Review PowerPoint 6. I'm going to delete that slide right now. All right, guys. Well, have a good day, evening, night, morning, wherever you're at. Bye.